what I've been doing for the past couple of years. So I come from the Department of Parasitology, Universiti Malaya, Malaysia. So we work, my laboratory extensively work on gut microbiota. So as you all from the healthcare business, we know gut microbiota has found a lot of traction in not just physical health, but also the mental health. There's a lot of studies and implication of gut microbiota in mental health, and there's a lot of things where gut microbiota has been found not just as a correlation, but as a causation already. For example, blood pressure, high blood pressure, we see gut microbiota is moving from a correlation to causation effect. So in that context, let me give you a few key facts about gut microbiota. So just to let you know, we have 10 times more bacterial or more microbial organisms in our gut compared to entire number of cells in our entire body. So that means we are only becoming more human when we pass our bowel in the morning. That's quite an important fact, because whenever we say me, we are to the point of 0.1, because we have 10 times more bacteria. And the genes, number of genes that humans have, it's about 22,000. And the amount of genes that from the external environment that influences us is millions of genes from the gut microbial organism. So we have a pertinent question to ask now. Now we have gut microbiota, and in the presence of certain parasites, what is the interaction that happens, and what is the implication of that interaction? So we asked this important question: can a harmless organism be turned harmful? Is there a role for gut microbiota here? Excuse me, sir, can we have it full screen? There's the 2 arrow button on the right hand top. You can click on that. Next one. Yes. Try again. Yeah, better. Thank you. So, so that was a question we asked. So, as I was talking about earlier, the gut microbiota. You see, I believe all of us know gut microbiota, and I also believe these two terms are often used interchangeably: microbiome and microbiota. Just before I delve deeper into that, microbiome refers to the entire genetic content of microorganisms found in any part. If you're talking about gut, then it's in the gut. Whereas microbiota refers to living cells. So, many researchers, I see that they often use these terms interchangeably, but this is the meaning, that they have different meaning. And this gut microbiota composition in the gut are significantly influenced by the diet, lifestyle, and also environmental signals such as the weather, climate, and also the medication that we take. And they influence, to a large extent, most of our organs. And systematically, we are significantly made up by our gut. And that's why our ancestors say that all diseases begins from the gut. Right? So, something that is often underlooked is the single-cell eukaryotes that are also a part of the gut. And my focus here is on the protozoa. See, these organisms, the protozoans, are often left out simply because they are 90% of the time asymptomatic. When they're asymptomatic, our medical professionals, they don't take they don't take any attention to it. But the question that we researchers should be asking is, why is it asymptomatic? What makes them to be asymptomatic in some while symptomatic in another? So that's the question we started off with. And we found that certain parasites, they have the ability to change the gut microbial balance. So in some cases, certain parasites in their presence, they are able to increase the microbial diversity, or in some cases, they can reduce the microbial diversity, both of which is not actually good. They have some implications. And the influence of certain parasite genotype, whether a specific subtype of parasite is the one that is causing damage while the other subtype is not going to cause any damage, is something that's not clear yet. But what is more important to see is that the interaction between them. So the impact of gut environment on parasite is something that is not properly studied yet, and that's where we are actually heading in. So in most of research that happens in my lab, we look at this organism called blastocystis. It's an intestinal protozoan organism, and why we picked this is simply because of their hyper-prevalent status. So they are super prevalent that no matter they're symptomatic or asymptomatic, you can still find them there. So, and this protozoan is often associated with non-specific gastrointestinal symptom, and more than 1 billion people were estimated to be infected in 2013. That was like about 12 years back. And now with the increase in emigration, we are probably going to expect more of this. And the prevalence is about, it depends on whether it's a developed country or developing country. And like I said, the prevalence is staggeringly high in most places. There are some studies which has reported 100% prevalence. The entire community has this organism, and it is able to colonize both symptomatic and asymptomatic. So we picked blastocystis as our model organism to test whether an organism, a harmless organism, can be turned harmful or not. And we leveraged on certain natural experiments. So in our first experiment, we isolated stool samples from the indigenous population of Malaysia, the Orang Asli population, and also we compared that with urban population. Both of them reportedly have significantly different gut microbial profile. They have different gut microbiota. And when this parasite has been isolated from these two different gut microbiota, how are they behaving, like how is their phenotypic characteristic? And we found that their growth was significantly different, and their apoptosis level, their self-death thing, which is a measure of pathogenicity that is often associated with blastocystis, and also their antigenic content. How do we test the antigenic content? Is whether they are able to proliferate or inhibit the growth of cancer cells. So they were all different. I'm not going to go into the depth of this technicality, it's too much. So they were different. Then we thought, okay, it could be because of the two different populations that we are comparing. What if we just focus on urban and we look at the specific disease state? Then we focused on another study that we were doing on schizophrenia, gut microbiota and schizophrenia, and we happened to have quite a number of schizophrenic patients who were infected with blastocystis. And we collected this organism from individuals with schizophrenia and individuals, other controlled individuals, non-schizophrenic individuals. And we found that again, that they are significantly different. They have different resistance to drug, and they released proteases in a different rate compared to the other group. So then we thought that, okay, there is something serious that's happening, so we should experiment more on it. So we did our own experiment where we collected samples, clinical samples from symptomatic patients from the same healthcare center, just to avoid any bias. And generally, an asymptomatic cells will have a growth profile as this, and asymptomatic cells will have a growth profile like this. But when asymptomatic parasite were grown with a bacteria from a symptomatic individual, we found that the parasite behaved like a symptomatic parasite, whereas vice versa, when the symptomatic isolates, parasitic isolates were grown with the bacteria from asymptomatic, they grew similar to asymptomatic. So what we did was we just swapped the bacteria, and we found that the parasites corresponded with the type of bacteria that were there. So we are now currently characterizing the bacteria that is there, and we are going deeper into omics, transcriptomics, and whole genome sequencing of the parasite, and to look at what are the genes that are being affected as a result of this parasitic changes in the parasitic growth and development that we are seeing in these experiments. But something important to remember here in the healthcare business is that the moment when you have a parasitic infection, we are immediately, we are told that it has to be immediately treated, given treatment. But this is opening a new venture, a new avenue that not all parasitic infection needs treatment. It then again depends on what type of interaction they have in there. Because the question that I started with is, can a harmless organism be converted into a harmful organism? It looks like it is likely based on some preliminary research that we have did. We have to do more to understand deeper. But this concept was already established in the damage response framework of microbial pathogenesis in 2003. That was like about 20 years ago by Casadevall and Malaya, and they said that pathogenicity is not a microbial trait but an outcome of interaction with its host. But they are not the first one. It was probably even our ancestors have already said that. Normally our grandparents or any learned one in the community, they will always say, you tell me who are your friends, I'll tell you who you are. So that's probably happening here, and that's what in fact is influencing any organism to exhibit or demonstrate their pathogenesis. So that's all. Thank you. I'll be happy to take some questions.
Clinically, how do we translate it when we are seeing patients? So we get patients with a lot of GI problems.
Yeah.
We may not know whether it is parasitic infection, whether it is I don't know when we will call it symptomatic or asymptomatic gut microbe.
Yeah. So that's the challenge we have now, because there's few factors that are involved here. One is the resource. Do we have resource to actually undertake all this experimental, because you have to do extraction, sequencing, there's a bit of, and most tertiary healthcare facilities are now incorporating all these sequencing facilities, next generation sequencing facilities in their hospital itself, because we are moving towards precision medicine and all that. But in resource limited setting, most of these things are often, there's a stark difference there. So that is, if you ask me, it's quite a challenging thing to translate at this point of time. But if you see 30, 40 years ago, it's rare for hospitals to have a PCR machine, polymerase chain reaction machine. Back then it was the thing that it's sort of a high technology. But now I see even most hospitals, most diagnostic labs have got a PCR machine, and they have a lot of molecular diagnosis that's ongoing. So we just have to wait and see. All right, so there's no question. Thank you.